it's not going to hold forever, so it's permanently like that, and that's no going to respond no matter what you actually no, I mean,
They recover, but yeah. Well,
the I mean, I'm not you're on the side. I don't get I love that basically last
Excellent job, Matt. So I am also Matt. This is Nico.
This is the Matt everyone liked at Recruit.
And we're here to introduce our wonderful housemate and good friend, Georgia Squyres. So everyone knows that Georgia is an outstanding scientist, so we're looking for some more obscure details for this introduction. So we'll start with her last name. So it has a very unique spelling, and this results in some very unfortunate mispronunciations. So we have a story that's loosely based on some true facts that will hopefully ensure that this never happens again. So this takes place many years ago in a magical land called Texas. There lived a family that was named Squyres. These thieves stole various items, including but not limited to cute and cuddly kittens. The problem was that George's ancestor, who was a well-known and well-respected judge, also was named Squyre. And she was sick and tired of being associated with this band of thieves, so she changed her name from Squyres with an I to Squyres with a Y. And so now the accomplishments of George's family would not be tarnished by the thieves that look oddly familiar to Tom.
So let's take a brief look at what else Georgia's family has done. So this is Stephen Squyres, Georgia's uncle. He has a PhD in astrophysics, professor at Cornell, and he was part of the Spirit rover and Opportunity rovers, which are two rovers currently cruising around on Mars. No big deal. He has won the Heresy Urey Prize, which sounds really awesome. He's the recipient of the Carl Sagan Award. We all know Carl Sagan is awesome. Plus he has his own Wikipedia page. Oh yeah, this is also Georgia's uncle dressed up in some fancy wannabe spacesuit doing who knows what under the
Pretty cool. Okay, so here's Tim Squyres, so George's father. Also mildly successful. He's a film editor. He's worked on films Unbroken, Life of Pi, a personal favorite, Crouching Tiger, Hidden Dragon. And yeah, he also took Nico and I out to dinner. So anyone who does that is successful in my eyes.
And then there's Georgia. So we try to find pictures of Georgia on Facebook because that's usually the go-to of what to do, and we weren't very successful, so we got Tina involved. Tina is Georgia's mom. Thanks to Tina, we found baby Georgia. Easy to recognize her, the red bright hair. And it looks like she's reading a book at a very early age, a book on fluffy guinea pigs and rabbits.
This is my personal favorite. Check out Georgia. So maybe we can zoom in or something. Oh, there we go. Okay. So Georgia is a very talented musician. She played piano at a young age, and she was in the drum line in high school and continued at Columbia. She also has great taste in music and an impressive knowledge on hip hop and R&B.
Quickly back to this. Also another fact from Tina. Georgia was the only girl in drumline for a really long time, and whenever they saw another drumline that had girls in it, they called them all Georgias. There are a bunch of Georgias drumming around somewhere. When Georgia was little, she also had two obsessions. One, using big words that no one understood, and two, penguins. Her penguin obsession was so big that one day she wanted to go visit penguin headquarters, which is right here. And imagine Georgia took off her orange life vest, turned around, she would fit right in. She also wanted to follow in her dad's footsteps and become part of the film industry. So he found a little clip of her on Nick News from a long time ago.
And listen up and pay attention, you might see something.
Yeah, there's a special hidden Easter egg. I really hope the sound works.
Now I realize it's possible for politicians to have their own agendas. And I've lost
No. Of course, it was our very own G1 Leo who was whispering anti-U.S. government sentiments into our audience.
But notice how Leo is secretly drinking an American beer hidden under his cloak.
So Georgia's rebellious phase continued into her undergraduate studies at Columbia, and yeah, we found this little article. We won't go into too much detail, but the title is "Columbia Mourns Departure of Four Loko." For those of you who don't know what Four Loko is, it's a beverage that does this to you. And Georgia was quoted as the Minister of Propaganda. Very impressive. Yeah, so Georgia came here, and I had the fortune of being in a mathematical modeling class with her, where she actually finished at the top of the class, and her project was featured on the website. Yeah, so she earned the title of MATLAB Queen. Yeah. Great. So all in all, Georgia, you're pretty cool. We're honored to have you as a colleague and friend, and I'm sure you're going to do great. So, that's it.
Now I see why my mom kept texting me about this talk. Now I see why she was there. Okay, in case you missed it, my name's Georgia, Squyres with a Y. And I'm going to start my talk out today with a scenario that I'll stress is completely hypothetical and in no way based in reality. So let's imagine that you are an MCO recruit and you have a really good time at some of our recruitment events, which is good, that's exactly what we want you to do. But then the next morning, for reasons that you can't even imagine, your stomach is really, really bothering you. And this is actually such a big problem that later when you interview with Florian Engert and he asks you about the moon phases, you draw a total blank and can't think of anything smart to say at all. So being a tech-savvy 21st century recruit, you go on WebMD to try to figure out what could possibly be the cause of your serious condition. And oh my goodness, you have cystic fibrosis, or some kind of cancer, Ebola. There's some kind of really serious problem going on. But there is a real lesson to be learned here, other than the fact that you should probably see a real doctor when you have a medical problem. And that's that, in fairness to WebMD, even for non-web browser-based physicians, it's very difficult for us to diagnose diseases of the gut. There are a couple of reasons for this. First of all, the techniques that we have to actually assay what's going on in somebody's gut at any given time are pretty inadequate and also tend to be really invasive. They're not fun. You don't want to do that. Further complicating things, we now realize that inside of every one of our guts, yes, even yours, there are on the order of 1,000 bacterial species that constitute the gut microbiome. They include maybe some of your favorites from the lab, but also a lot of bacteria species that are very obscure to us, and that's because the majority of these species are actually unculturable. To make matters even more complicated, they interact with each other and with us in very cryptic ways, but we do know that they have very serious impacts on human health, including our propensities for obesity and cancer and infection. So if we now imagine what a best case scenario would be for trying to figure out what's going on in our guts and why we flubbed our interview so badly, we could imagine if we could take one of these gut bacteria and convert it into a spy for us. We could send it into the gut where it would blend in flawlessly with its environment, take some observations, and then report back to us on what it's seen. So that's actually what we're going to do today. So because we're very diligent graduate students, let's make a to-do list to keep us on track in the lab. So the first thing we want to do is actually make this bacterial spine. Then we want to put it into the gut, but we're biologists, so we'll put it in the mouse's gut first. Then there's probably some other stuff we need to do at some point, but ultimately we want to use this to help people and hopefully make a little bit of money. So the technology that we're going to use to build our spy bacteria comes from a field called DNA computing. For those of you who aren't familiar, it's actually conceptually pretty simple. So in the same way that with hardware computers, you put in some amazing MATLAB code, and you pray that what you get back out is good enough to earn you an amazing grade in MCP 111, DNA computers have molecular inputs and outputs, and all of the computational and logical processes are done by DNA itself and its gene products. This field was originated by some nerd named Leonard Adleman in 1994. I bring him up because he's in fact not a biologist at all. He's a hardcore computer science guy. And his involvement explains a little bit about why the field evolved the way it did. So when people were first talking about DNA computing, they were interested in using DNA to augment or supplement or even replace hardware computers for specific types of computational problems. So some of the first big work done in DNA computing was actually using DNA to solve computation problems. So the traveling salesman problem is sort of a classic proof of principle in computer science, and people solved that with DNA a little while ago. Now people are very interested in the possibility of using DNA as a storage medium. So a British group a couple years ago encoded all of Shakespeare's sonnets in DNA. George Church naturally distributed his book on DNA. And now we're actually able to store 700 terabytes of information in a single gram of DNA. Just to give you a sense of how impressive that is, if we wanted to store 700 terabytes on conventional hard drives, you need to bring a really big hand truck because that would weigh about 151 kilograms combined. So this is really a lot better than anything we can make, and people are still very interested in this. But what we're going to talk about today is what happens when you give all these DNA circuits to a synthetic biologist. And that's the idea of making these programmable bacteria. So we take all of the DNA circuits made by Leonard Adleman and friends, put them into a bacteria, and then we have bacteria that we can program to carry out all of our nefarious tasks for us. So hopefully I don't need to convince you guys that this technology is really cool. So this is Pam Silver, in whose work, in whose lab the work I'm going to talk about today was done, and as you can see, she's very cool. But she has this wonderful quote about the fact that being cool is not enough for her, and it should not be enough for you either. So she says, "We're tired of making toy systems that don't necessarily have an application." And I think this captures both her motivations for doing this work and my motivations for presenting this paper, which is that DNA computing has been around now for 20 years. We have these DNA circuits that do increasingly cool and complex things, and yet a lot of what we've been doing with them is encoding Shakespeare's DNA, stuff that isn't necessarily directly relevant, particularly to human health outcomes. And so this is a paper that really strives to bridge this gap between this technology and an outcome that's really relevant to us. So the paper I'm going to present to you guys today is called Programmable Bacteria Detect and Record an Environmental Signal in the Mammalian Gut. It was published in PNAS in April of last year, and the work was done at the Wyss Institute here at Harvard and Silver. So I just told you guys that we were going to be using DNA circuits to program our bacteria. There are a ton of different types of DNA circuits, which I'd be happy to talk more about at the end if you guys are nerds. But the one that we're going to focus on today is a memory circuit. And it turns out that memory circuits are particularly well described by this very basic example of a button and a light. So when we start our super rigorous experiment, this light is off. Then we touch the button. The light turns on, and then even when we take our finger off the button, miracle of miracles, the light stays on. Now I bring this up not because I don't think you guys know how to use a light, but because this example actually captures two really important features of what we want our memory circuits to do. So first of all, even though we're no longer pressing the button, we have walked away to get a cup of coffee, this light remains on. So then we can come back later and say, oh, the light's on, somebody's pressed the button. So that's what I mean when I say a circuit that can remember. Second of all, we don't want, as we do the experiment, this light to be blinking on and off without any input from us. Not only would that be incredibly annoying, but you can see how that would confound our ability to read out reliably what this circuit is telling us. So we need both the on and off states of the light to be super stable. So we're going to call this biostability, and that's another requirement that we have for our memory circuit. Now the best part about being a synthetic biologist is now that we have some sense of what we want to make, we can actually go into nature and try to scavenge something that evolution has tuned to do exactly that thing. So in this case, the hero of our story is the lambdaphage. Lambdaphage is a very well-studied bacteriophage, so I'm showing it here infecting a bacteria. And as soon as it makes this infection, it has a really important decision to make. even bigger than where to go to finish graduate school. And that's which of these two pathways it should take. So if the bacteria it infects is healthy, it's sweet, it incorporates its own DNA into the host genome and then waits. And the advantage of this strategy is that now it's infected not only the initial host bacteria, but also all of its progeny. On the other hand, if it infects a bacteria and the bacteria is unhealthy, conditions are bad, it says, whoa, this is no fun, I should make a bunch of copies of myself and GTFO. And that's exactly what it does in the lytic pathway. And, surprise, surprise, this decision is made and then maintained by a genetic bistable switch, which is what I'm showing you here. So there's actually two genes involved, it's a very simple system. So the C1 gene regulates lysogeny and the CRO gene regulates lysis. And they mutually inhibit each other's expression. So we'll call this a double negative feedback. And actually just having this double negative feedback alone is sufficient to establish a system with two bistable states. As you can see, there's one where C1 gene expression is high and one where CRO gene expression is high. So now that we have found in nature this amazing bistable switch, all we need to do is rip it out of the lambda-phage and put it into what we want to make. So that's what the authors do. So here's the synthetic memory circuit that the authors build in this paper, and you'll see that the memory element down here, all of this is taking pretty much whole cloth from the lambda-phage genome. The one big modification that we've made is that after this CRO gene, we've added a reporter construct. That's just so that we, the experimentalists, can read out what state this memory element is in. The other thing we need to do here is to make it so that the state of our memory element is responsive to something that we want to detect in the environment. In this paper, we're going to be detecting tetracycline. So the way we do that is we build the second sensor element, where we have a second copy of the CRO gene under the control of the tetracycline-inducible promoter. So the way this works in the course of our experiments is like so. At the beginning of all of our experiments, we start in the C1 state. That's kind of the default for this circuit, and I'm going to call this the off state from now on. This sensor element up here is, of course, also off because there's no tetracycline present. Then at some point during our experiment, we apply our stimulus, so we add tetracycline. You can see that turns the sensor element on. We start to express CRO here. That represses C1 expression, which in turn de-represses CRO expression. And so now, even after we remove tetracycline, this CRO gene has turned on, and our memory element has stably flipped into the on state. So if we come back later after lunch and see that now we have our reporter gene being expressed, we know that this circuit has been exposed to tetracycline in the past. So now that we've designed our memory circuit, it's time to put it to the test. So the way we do that is first we put our circuit into a bacteria. So this is just a standard lab strain of E. coli. And so what the authors do is they grow this bacteria in culture with tetracycline for 6 hours. And after 6 hours, they screen to see what states our bacteria are in, just for looking by looking at the reporter. And they see that 100% of the bacteria have switched into the on state. Then they take tetracycline out of the medium, wait 5 days, and screen again. And they see that even after 5 days, 91% of these bacteria are still in the on state. So that tells us a couple of things. First of all, our circuit actually can sense tetracycline. Also that this conversion to the on state, this initial sensing is very robust. We actually have a 100% conversion after 6 hours of exposure, which is pretty darn good. And then also that our on state is stable for quite a while. So even after 5 days, the great majority of our bacteria are still on. So there's a good deal of stability to this on state. For a control, they do the exact same experiment without adding tetracycline, and you can see that for the entire duration of the experiment, the bacteria stay off. So our off state is also really stable, and in fact, there are 0 false positives from this experiment. And for every experiment we do thereafter with the circuit, there will continue to be 0 false positives. So particularly this C1 state turns out to be incredibly stable. And we have evolution to thank for that. That's not us being smart. Which is cool. Before we move on from here, there's a couple of numbers I want to point out to you guys. So the concentration of tetracycline it takes to flip the switch is about 100 nanograms per mil, so pretty reasonable. And at that concentration, it takes about 3 hours for these bacteria to switch on. I bring this up not because I particularly want you to remember those numbers, but just because I want you guys to already be thinking ahead to potential applications. Because I told you the object of the game is to come up with an application for these technologies. So it should already be reassuring to us that these aren't egregiously large numbers. So now, I think we can go ahead and satisfy ourselves that we've had a productive morning. We've made our spy bacteria. So now it's time to go ahead and move into a mouse. So we do that like so. So we feed our spy bacteria to a mouse. Then we expose the mice to tetracycline. You do that just by adding tetracycline to their drinking water. Then you wait a couple days. Then you collect a sample of our bacteria back from the mouse's gut flora, which you do exactly how you'd expect. And then screen these bacteria to see what state they're in. So first of all, I did just say that we put spy bacteria into mouse, get out from mouse gut. That's not a totally trivial thing to do. So the way you do this, what I'm showing you here is the bacterial population graph. Notice the log scale on the y-axis. In black is the mouse's native gut flora. Now, I did tell you at the beginning of this talk that more than half of those are not culturable, and that was not a lie. This is not the entire gut flora of a mouse. This is just a proxy of what we can culture under these conditions. So this isn't a raw number that you should take home about how many bacteria are in a mouse gut. This is just a proxy that tells us roughly what's going on. And then in yellow is the spy strain that we're adding. So at the very beginning of this experiment, what we have to do is dose these mice with a really, really, really large dose of streptomycin. And what that does is that actually gets rid of the great majority of their native gut flora, as you can see by this sort of stat graph point down here. That's necessary because if we didn't do that and we just fed our bacteria to mice, they would have no space in the gut niche to colonize. So we need to wipe out a bunch of the native gut flora so that our bacteria can actually grab a foothold. And you'll see that after we do that, and we feed our mice these bacteria, we see them in their gut for a while. So now that we've established that we can put them in the gut, it's time to go ahead and do our experiment. And this experiment ends up working actually really well. So once we put our bacteria into the gut and we expose the mouse to tetracycline, 100% of these bacteria flip into the on state. And then after we remove tetracycline, even a week later, more than 60% of them are still on. And then if we compare that to the control, where the mice are not exposed to tetracycline, again we see that this experiment has no false positives, which is nice. So we see that this is working just about as well in the gut of a mouse as it is in culture, and that we can actually, just with this bacteria, tell whether or not the mice have been fed tetracycline, which is pretty cool. But we're perfectionists, and if we look back at our to-do list, we can see that we did want to colonize the mouse gut. And this graph is not really colonization, it's actually a little sad. So this is a really steep drop-off after we feed these mice our bacteria, and actually not too long after this time point, they'll be totally gone from the gut. If we want to move into humans, or use this to make some sweet profit later, clearly this is not going to cut it. But let's not be too hard on ourselves. We shouldn't be that surprised by this result. What we've essentially done is we've taken a strain that was evolved to survive in the lab, put it in the mouse gut, and expected it to do just that. It really makes no sense. So it would be like if we took G1 Matt Riley, who evolved in Portland, Oregon, put him down in the middle of Paris and expected him to survive, when in reality, he maybe has some very unfavorable interactions with the locals and dies out. So we should think about pursuing a new strategy, where instead we take an organism that did evolve in this environment, bring it in G1, we evolved down, and then maybe we would think that things would go differently. His interactions would be a little bit more favorable, or maybe stably colonize this environment. So what that means for the authors of this paper is that they're going to go into the gut of a mouse, try to isolate a bacteria that's evolved to survive and thrive there, and then redo all their experiments in this new bacteria. So first, they collect a sample of natural gut flora from the mouse, again, probably exactly the way you're expecting. Then they screen through to look for a bacteria that's similar to E. coli. They do this because they did all their initial work in E. coli, so you might as well try to be as consistent as you can. So first they screen for bacteria that are gram-negative and lactose fermenting, which E. coli. And then second, they sequence the 16S ribosomal RNA. That's a standard in bacterial identification and phylogeny. And you can see that they're able to isolate a natural strain that's very similar both to our original lab spy strain and to their reference E. coli as well. And then we take our whole memory circuit and move it into this new bacteria, which I'm going to call the natural spy. You can see he has a beret on. So things are Okay. So the first thing to do is make sure that our memory circuit works as well in this new strain as it did in our original lab strain. So this is the mouse data that I just showed you before. And remember that our original lab strain is in blue. And now I'm going to overlay in green our new natural spy strain. And you can see that this really does just as well as our original lab strain. This is encouraging to us for two reasons. First of all, it means that yay, our experiment worked and we can move forward with this bacteria in the future. But second of all, and sort of more generally, it means that if we want to do things like this later, where we are designing synthetic DNA circuits to be used in a natural environment, we can build, optimize, troubleshoot those circuits in strains that we already have in the lab, your lab mate has in the freezer, you're very comfortable working with, and then as a final step after all of that work is done, move them into a natural strain that's similar and expect them to work just as well. But now the moment of truth, we can ask ourselves if this is any better at surviving in the mouse gut, and I've kind of given it away, but I'll walk you through the graph anyway. So again, this is bacterial population in the gut. Our original lab strain is in blue. Our new natural spy is in yellow. And again, we just have a reference number for native gut flora in red. And the y-axis, again, is on a log scale. So just as we saw before, so too do we see here, that the lab strain is sadly not able to colonize in the long term and quickly dies out. By contrast, it seems like our natural spy is doing much better. So it seems like we've almost hit a stable number here for colonization. The authors do claim that this is going to be stable forever. I wish they had had a day 25 or 50 or 100 time point on this graph to really convince us of that. I think it would be really funny to have a day 100 bar here, but I think it would be more convincing than these two. But either way, it's certainly doing better than our original lab strain, so I'm going to go ahead and say that we have successfully colonized the mouse gut. So that actually brings me to the end of the paper I wanted to talk to you about today. It's not a bad day's work in the lab, but we should probably think about what's maybe next for this technology. I'm sure you guys have a lot of ideas, but I'll talk to you a little bit about what I thought was potentially the most interesting. So I think maybe the most obvious thing to think about is using this to detect something that's actually interesting to us. This whole paper we've been detecting tetracycline, which is great for proof of principle, but obviously is not what we're looking for in the real world. We want to detect cancer, we want to detect pathogens or toxin exposure. So tetracycline is not going to cut it. So for our particular circuit, what that means is we need to swap out our tetracycline-inducible promoter for a promoter that's induced by something that we actually care about. So there's actually a reasonable host of these promoters already in nature. For pathogens, any pathogen that produces quorum sensing factors will probably also have a promoter that's inducible by those factors. That's kind of how quorum sensing works. So Pseudomonas aeruginosa, Vibrio cholera, those sorts of bacteria will probably all do have inducible promoters. And there are other pathogen-inducible ones as well, for some reason, especially in plants. If there's any plant biologists out there, get hype right now. For cancer, there's a hypoxia-inducible one that people are interested in for tumors, and there are a lot of others I could list for a while. My point is there are actually promoters that we already have in nature that could be very relevant for this. So I don't think I'm crazy when I say I've gone ahead and imagined what that public health decay might look like if we use this technology diagnostic. If your poop flows, you should probably go to the doctor because you potentially have a very serious disease. Something else that the authors actually talk about as a possibility is using these bacteria to not only diagnose a disease, but then to go on and administer a treatment. So in our system, in the on state, we've just been expressing a reporter so that we can tell what's going on. But there's no reason that in addition to or instead of that reporter, we couldn't also express a therapeutic. So particularly in the case of pathogens, bacteria have been waging bloody war on each other for millennia, so a lot of bacteria actually already make antimicrobial peptides to kill each other that we can express. So there's an example of a 2013 paper where they do search and destroy bacteria in Pseudomonas, which I will going to talk about more if you guys are interested, but certainly this isn't completely far-fetched to imagine. And then we can actually update our public health campaign to say that if your poop flows, you're good to go. Because you're being treated for whatever serious disease. And then as just a final point, I've been speaking this entire talk like the C1 Pro memory circuit is the only one that exists. And that's completely untrue. It's not even the best studied one. And again, if you guys are interested, I'd be happy to talk about what the alternatives are and why the authors chose this one specifically. But for now, there are actually a lot of different memory circuits out there, and those different circuits have very different properties. And I think something that's really interesting to consider is the idea of choosing a specific memory circuit based on the application that you wanted. So an example that'll maybe make that clear is if we think about our circuit, we know that the on state is stable for maybe a couple weeks, but it's not stable forever. If we wanted to use this to do diagnostics, and we wanted to screen patients once every 6 months, obviously a 2-week on state is not going to cut it, and we would need to look for a circuit with a more stable on state. Those circuits actually exist. On the other hand, if we wanted to use these to diagnose and treat a pathogen, it might be nice to have something that stays on for a while and then shuts back off again. If you have a Pseudomonas infection at age 15, you don't need to be given antimicrobial for like the rest of your natural life. 2 weeks is fine. So in that case, this on state might actually be perfect for an application like that. And I think in particular this example makes me think that this technique that they've outlined here is applicable more broadly than the examples I talked to you about today. But on that note, I'm going to end and say a huge thank you to my coaches, Vlad and Elena, who were incredibly helpful, and my fellow speaker, Matt Riley. And I'll take any questions. idea of having a circuit that you could turn off. So actually, that wouldn't be that hard to implement with our circuit. So if we had a second sensor with a second inducible promoter that drove C1 expression, you can imagine that we could tip the scale in both directions. There are other recombinase-based circuits where you actually can have multiple switch mechanisms where you can turn things on and off as well.
So,
Yeah, go ahead.
in the
Sorry.
experiment, technically, how to make sure that we have removed the inducer 100%?
So the authors actually screen both in culture and in the mouse, and there's no tetracycline detectable after 6 hours. So they're very confident that, especially a week out, there's no more tetracycline in the mouse system. That's your question.
So this methodology always required like evacuating and killing all the normal gut flora first?
No. So this is a protocol developed for mouse, and in mice, obviously, we can do this. But in humans, killing all of your natural gut flora is really not something you want to do. But we've already seen people develop therapeutics that involve gut transplants, and what you're doing there is transplanting fecal microbiota. So yes, it's possible to do this in humans without dosing people massively with antibiotics. Thanks, guys. Enjoy your pizza and beer.